Hello and welcome to this channel. Today's press information. A new study suggests that a previously hidden reservoir of HIV may be destroyed by using mRNA vaccines. This would bring us significantly closer to a cure for AIDS. Let's get started. As we know, HIV infection leads to acquired immunodeficiency syndrome, AIDS, by destroying the body's immune system. Although highly active antiretroviral therapy, HAART, has allowed people living with HIV to live longer and healthier lives, it does not actually cure the disease. Because the virus can hide within certain white blood cells called T cells, it cannot be completely removed from the body. This viral reservoir makes it possible for HIV to reactivate even if you're taking medicine to prevent it. Currently, the only way to prevent HIV from reactivating is to take antiretroviral therapy for the rest of your life. And unfortunately, almost 40 million people worldwide are living with HIV and need to take these drugs every day. One major challenge to curing HIV is that it can hide in a part of the body that is difficult to get to, but this new discovery might change everything. The team behind the new research sought to discover a way to access the parts of the body where HIV hides in order to remove it. To do this, they took advantage of a strategy that has been successfully used against other viruses, namely to instruct the immune system to identify and destroy the infected cells. The study authors say that using mRNA technology, which was first used in the COVID-19 vaccines made by Moderna and Pfizer-BioNTech, could be key to making this happen. Specifically, they wanted to use modified mRNA molecules enclosed in tiny fat bubbles known as LNPs to carry the genetic instructions. According to the researchers, although mRNA has been used to develop effective cancer immunotherapies, it has not been used in the fight against infectious diseases like HIV. They also noted that previous attempts to use mRNA to treat HIV have failed because the cells where the virus likes to hide, specifically memory CD4 T cells, don't take up the fat bubbles or LNPs that carry the mRNA. In a paper published in Nature Communications, the researchers showed for the first time that they had developed a way to make the virus visible by encapsulating the mRNA in a newly designed type of LNP. This delivery system allows the virus to be revealed inside the body's reservoir where it is hiding, which would allow the immune system to finally reach it. Their new design is known as LNPX. Our hope is that this new nanoparticle design could be a new pathway to an HIV cure, said Dr. Paula Seval, a research fellow at the Doherty Institute and co-first author of the study. To understand why this is such a big deal, let's look at how HIV typically evades the immune system. HIV attacks specific white blood cells called CD4 T cells, which help coordinate the body's immune response. As the virus replicates and destroys these cells, the body's ability to fight off infections and diseases is weakened. The virus attacks the helper T cells directly, causing them to die and gradually destroying the immune system over time. By the time the immune system is severely damaged, the person develops AIDS and is susceptible to life-threatening infections and cancers. Normally, the body gets rid of viruses by producing antibodies against them. However, HIV has a clever trick to avoid this defense mechanism. The virus has an enzyme called reverse transcriptase that allows it to convert its RNA into DNA, which can then integrate into the DNA of the host cell, allowing it to hide from the immune system. The problem is that once integrated into the genome, the virus becomes very difficult to target with traditional antiviral drugs. However, the researchers think that instead of trying to block HIV's ability to integrate into the host genome, a better approach may be to target the infected cells themselves. If they can find a way to identify and destroy the infected cells, they may be able to clear the virus from the body entirely. One way to identify infected cells is to use mRNA vaccines. These work by introducing a small piece of the virus's RNA into the body. Once inside the cells, the RNA is translated into protein, which can then be recognized by the immune system. The mRNA vaccine instructs the cells to make a viral protein that is normally found on the surface of the virus. This protein acts as a flag, making it easy for the immune system to identify and destroy any cells that display it. However, there is a challenge with using mRNA vaccines to target HIV. As I mentioned earlier, the virus likes to hide inside certain white blood cells called memory CD4 T cells. These cells do not take up the fat bubbles or LNPs that carry the mRNA. This is a problem because without a way to deliver the mRNA to these cells, it's impossible to stimulate the immune system to destroy them. However, the researchers believe they have solved this problem with their new LNP design. In their study, they tested their new delivery system in mice with humanized immune systems. They found that their new LNP design was able to deliver mRNA to memory CD4 T cells and stimulate the immune system to destroy them. They also showed that their approach was effective at reducing the amount of HIV in the body. Now, it's important to note that this study was conducted in mice with humanized immune systems, so it's not clear whether it will have the same effect in humans. However, the researchers are hopeful that their new approach will lead to the development of a cure for HIV. According to Dr. O'Leary, the next step in their research is to conduct clinical trials to evaluate the safety and efficacy of their new treatment. If all goes well, they hope to have a cure for HIV within the next 5 years. Now, although this is only a recent discovery, scientists have been working hard to develop a cure for HIV for decades. One promising approach has been the use of gene editing tools such as CRISPR-Cas9 to remove the virus from infected cells. However, this approach is still in the experimental stages, and it's not yet clear whether it will be safe or effective in humans. Another approach has been the use of immunotherapy to boost the immune system's ability to fight off HIV. However, this approach has also had limited success. The researchers believe that their new approach of using mRNA vaccines to target infected cells is a promising new strategy for curing HIV. There are several advantages to using mRNA vaccines. First, they are relatively easy to make and can be produced quickly. Second, they are very specific and can target infected cells without harming healthy cells. And third, they are safe. There have been no serious side effects associated with the use of mRNA vaccines. I know that this topic can be quite technical and complex, so I hope that I have done a good job of breaking it down and explaining it in simple terms. If you found this video helpful, please let me know in the comments below. I always appreciate your support, and I'm happy to answer any questions you may have. That being said, it's important to remember that science is always evolving, and there is still much that we do not know about HIV and AIDS. However, I am optimistic that this new discovery will lead to a cure for the disease within the next few years. Until then, it's important to continue raising awareness and funding for research to find a cure. Thank you again for watching, and I look forward to seeing you in the next video.